what is the microbiome? Is our 100 trillion closest friends. They live in and on us. They are anything from bacteria, viruses, fungi, etc. And it is, as you have by now gathered, a pretty hot topic across medicine, actually, but particularly for brain disorders. And that has become particularly important and attractive since there are several mechanisms discovered how the gut and the microbes in the gut can actually communicate with the brain and vice versa. And so this connection can be established by several means, and we think this could be the immune system. Think inflammation, of course, as well. You heard about the inflammation. Anything that is body fluid, so for example, blood that can transport things like hormones, neurotransmitters, and metabolites. And then in a sort of hardwiring way with the vagus nerve, which connects the nervous system of the gut to the brain. So what are some of the conditions that are linked in some way to the microbiome? There's anything from closer to gut conditions such as inflammatory gut diseases, of course, obesity, depression, and anxiety, which of course are topics that are very relevant for Parkinson's disease as well. Pain, autism, multiple sclerosis, cardiovascular risk, all of these have been linked, and in the last 1 or 2 years, Parkinson's disease has been one of those conditions that are of particular interest. Why would that be? Well, from a clinical point of view, and that is where the clinician's view comes in, constipation as well as a poor sense of smell can actually occur years, if not decades, before anybody develops any motor symptoms of Parkinson's disease. Now, we just heard about the importance of figuring out what is happening before the symptoms make themselves known. So with that in mind, that the clinical symptoms affect the gut and the nose, Parkinson's disease might, at least for a good proportion of people, well start in the gut or the nose. Now, of course, those surfaces, these are lined by mucosa, so that is what we have as an internal surface. They are very densely populated by microbes. And it is only now that we're really trying to understand and starting to understand this vast, vast population of microbes and what they're doing. In addition, alpha-synuclein, which is what you've just heard about, which is the major protein that is clumping up and clogging everything in Parkinson's disease, can not only be found in the brain, but also in the gut. That is not a one-to-one relationship, and there's some questions on if you have that protein in the gut, whether that definitely means Parkinson's or not. But suffice it to say, for now, there is alpha-synuclein in the nervous system of the gut, and it has been shown in principle that it can travel from the gut also to the brain. Now, fortunately, the microbiome in the gut, the mouth, and the nose is quite easily accessible. Brain samples are very difficult to come by, whereas poop samples are not. And so where are we moving to? What are the goals of research in the microbiome and in Parkinson's? So we, first of all, need to understand whether changes in the microbiome, whether variations in the microbiome are actually related to Parkinson's disease itself, as well as to potential subtypes. So think of, for example, anxiety and depression, but there are also certain motor subtypes of problems with balance, for example. And if that is the case, are these changes in the microbiome indeed a cause or part of a cause for Parkinson's? So first you have a correlation, and then you need to look into the cause. And what is the specific mechanism or mechanisms that provide this link? And how early can we detect any changes in the microbiome? Can we see changes before the motor symptoms set in? And then, of course, future studies, and that is now really very much down the line, would look at, can we establish a biomarker, or can we even establish therapies such as antibiotics, probiotics, etc. But before we can look at any of those really lofty goals, we need to do our homework and do the basics, and that is what I'm talking about today. And so what has been done in Parkinson's disease so far? There have been 4 all fairly small studies published. They all found significant differences, but the results are anything from homogeneous. So every study found something, but they didn't necessarily find the same thing. So we really need larger studies, and we need to have longitudinal follow-up of those. All of the studies that are published analyzed either feces, so poop itself, or mucosa samples from the gut. But none of them have so far studied the oral or nasal microbiome, which is what we're going to do. All of them analyze bacteria, but none of them have looked into any of the other species, such as, for example, fungi. And that is what we're going to do as well. So the microbiome project here that we're about to start, it analyzes the microbiome both in fecal samples as well as in oronasal samples. In both participants with PD as well as in healthy controls. You always need healthy controls. I can't say it loud enough. Without the healthy controls, we can't do our research. So anybody who is here and thinks, how can I contribute? If you're a healthy control, go for it. We're going to look for bacteria and fungi. We're going to look at a correlation of those changes that we are hoping to see with the presence of Parkinson's disease, as well as hopefully with subtypes, be that in the non-motor or the motor domain, and then of course with progression, so we can follow participants over time. We'll also collect blood samples so that we can, at this stage in an explorative way, also see, are there any inflammatory markers, for example, are there any metabolites that change in correlation with changes in the microbiome. So we have a very multidisciplinary team here at UBC. Of course, this is our movement disorder neurologist here, so it's coming from the clinic and it goes right back in the clinic and it happens in the clinic. But in the meantime, we then also have the neuroscience expertise of Dr. Brian McVicker, one of Canada's most eminent neuroscientists. He's the co-director of the Center for Brain Health, along with John Stessel. We've got also very eminent microbiologist, Dr. Brett Finley, on board, who has vast experience in that field. And then we've got us as clinicians, and thanks to your funding, not doctor yet, but Kristen Sandvik is going to do the project coordination. So with that, the very exciting news. So when I talked about this project last year, it was an idea, it wasn't funded yet, and since then I really thank you for funding the project so that we can go ahead, the pilot project. And since then, more good news. So we applied for a pilot project funding from the Parkinson's Society of Canada. That was the competition of the Canadian Parkinson's Society, so countrywide. And we got the pilot project funding awarded, and then very graciously the Parkinson's Society of BC stepped in and said, yeah, we are going to sponsor the project, so thank you very much there as well. CBH is providing matched funding for the project as well. And then later on, that is very recent news, we then, because of the expertise here, together with Dr. Brian McVicker and Dr. Brett Finley, we also have funding to do this kind of research in a subset of Alzheimer's patients. And I think we can certainly learn from each other for the Canadian Consortium on Neurodegeneration in Aging, the CCNA. So in the meantime, several other funders, and including competitions for funding, have stepped up. So where are we now? So ethics is in place. Some minor updates are done at present, mainly to incorporate our collection with the biobank at CBH, and Dr. Stiss already mentioned we are going to contribute to the biobank, and the biobank in return is also going to help us with wet lab space. Hospital approval is in place. We've got packets of specimen collection kits, which are very interesting and hopefully easy to use. I mentioned the wet lab portion that is being sorted out right now and should be in place shortly. And then we already have the analysis pipeline in Dr. Brett Finley's lab. So next steps will be to enroll hopefully 75 participants with Parkinson's disease and 50 healthy controls. We're looking especially, but not exclusively, for spouses, as they would share diet and environment, their advantages and disadvantages for having spouses in the program. But I think at this stage, where we want to keep it as homogeneous as possible, that would be an advantage. But if you're not a spouse and you still want to contribute, feel free. Individuals should be within 8 years of onset of motor symptoms and should be between 40 and 70 years old. The reason for that is that the microbiome is expected to be fairly stable in that time, and it might change after the age of 70. In a follow-up study, we might look at all the participants as well, as this might well be relevant with Parkinson's as an aging condition. Yeah, but for now, to get a fairly homogeneous sample, this is what we're going to do. What is collected? So, of course, you know the usual clinic visit with Parkinson's history and current symptoms. We'll have questionnaires that look at any confounders. So that could be anything from diet to pets to mood to constipation. We've got exercise and occupation in there as well. We'll do a Parkinson's disease physical exam, so UPDRS, as well as a very specific smell test. Remember, we're collecting from the nose as well, a brief cognitive screen, and then we're taking these oronasal samples as well as blood samples in the clinic, and you'll be sent home with a kit to get a stool sample. Timeline. So in the next 12 to 18 months, we hope to enroll everybody, collect the samples, do the clinical assessments, and store the samples until the batch can be analyzed. That should then hopefully happen in about 18 months' time with the analysis of the results and writing up for publication, and of course sharing with you along the way, expected in the fall of 2018. And with that, I thank you for your very generous support and hand over back to Dr. Mack.
Thank you very much.